Oh, yeah, everyone, good morning, good afternoon, good evening as well. I'm June. As kindly introduced, I work in a Korea university, and my lab is basically working on the genetics of autism. And maybe some of you might know that the autism is really biased toward the males at the current research. So I'm going to highlight some our latest finding about sex bias and also sex difference in autism by we see in the genetic research. So in let's start with the very initial observation, not in autism, in other conditions. So this is the very early findings by Cedric Carter, a British clinicians. So he looking at one of the congenital stomach disease called pyrortic stenosis in early 1960s. He's found that the this condition is highly observed in boys than girls, and I mean that was quite interesting observations because there's some sex bias. But interestingly, the female patients they even though very rare, but their relatives have quite high in terms of the occurrence of this disease. And he thought that why males have more diagnosed towards this disease, but females are, less diagnosed than males. And also the occurrence is quite opposite in terms of the different sexes as well. So he observed that issue, and later on, other geneticists and other scientists also observed the similar things in other disorders like kidney stones and clubfoot, peptic ulcer, and cleft lip. And there was, becoming some generalized idea about what we call Carter effect, which is some disease or disorders as could certain sex, but also the rarely affected sex must carry higher genetic liabilities or other risk factors that transmit more risk to other relatives. So one thing we can think about is different threshold of being diagnosed by different sexes, and also different levels of risk factors affecting on each sexes as well. And these things is really clear in autism as well. So if you think about the prevalence of autism, we can say it's about 4-to-1 ratio. And the ratio is a little bit up and down across different studies. So very early observations by Leo Kanner in 1943, there was the ratio about 3-to-1, but now it's about 4-to-1 ratio. And also the ratio is quite replicated across different epidemiology and also different national data as well. And we're going to discuss about the ascertainment about this different sex ratio. But if you're looking at more conserved and also more active ascertainment, which means that we can looking up some, the sex or gender bias in terms of the ascertainment, that we can see the bit lower sex bias than the original observation, so which is about 3-to-1. But this is still we have higher sex ratio in terms of the male bias in the active ascertainment as well. And also interestingly, if you're looking at the different angles of this sex ratio, so if you're looking at the different functional levels or different the presence of the intellectual disabilities or the cognitive levels, we can see that about like a 7-to-1 ratio in terms of high function in ASD, or the 2-to-1 ratio in severe intellectual disabilities as well. So this is all the affecting on the sex ratio in autism research. So back to the Carter's observation. So if you're looking at the autistic males versus females, you see the male has higher the prevalence and occurrence, but their brothers or sisters way less affected than the female proben. So if you're looking at the female proben, they have way higher in terms of the brother affected and also sisters affected. So this is also indicating that there are some risk factors affecting on sexes in different ways and also different levels of contributions as well. So why we have more observations in males than females? So three different layers we can think of. So first one is we can think of the systematic levels, which is we can looking at the diagnostic criteria. So when we diagnose males or females in clinical settings, we probably see some diagnostic criteria preference to males, so that's why we collect more males in current system. Or in other layers we can think of is societal levels. So if you think about the males and females, the females they now know about the camouflaging. So if you in a situation where you diagnose for autism or in a social relationship, you have the females have the camouflaging the situations where they hiding there or they reluctant to the some situations in terms of the social difficulties. So that also one area people think about the gender differences in terms of this sex bias as well. And another layers we're going to discuss mostly on this presentation is about the biological factors. So as you know that the sex difference is also varying by the different levels of biological factors like genes, variant, and hormonal differences. All of these things also affecting on this sex difference in autism. So one idea people discuss in these days is the female protective effect, which is the levels of being affected by autism is differed by different sexes. So we have the different levels of the threshold and also different levels of the contributions by different risk factors. So the model, very simplistic way, we can say that the females require greater cumulative liability to cross the autism diagnostic threshold. So if you have a diagnosis for autism, females should have much higher risk than males. And that is widely observed by different levels of studies. So one is the genetic factors, so rare variant, which is very lower frequency in a population, which is great impact on the autism development. And these variants are quite excessive in females than males. So that is one very strong evidence for now. And different one is that the polygenic background as well. So that also has the higher impact in females than higher contributions to higher level of the polygenic background in females than males, but the impact is quite sensitized to the males. So that is another things. And also another evidence we can think of is the epidemiology evidence. So as we discussed in previous slides, so Carter effect like observations we see in the sibling recurrence as well. So that is also the things we can think of. And also female protective effect is actively working in terms of the neurobiology of autism. So how these things is really affecting on the different the biology of the different sexes. So the neurons or microglia is working on the very early development of the especially like a fetal brain stage. So all of these ideas we also lie with the Korean context as well. So since I moved to Korea in 2019, I've been actively working on the research of autism genetics. So as I study in other countries before my the time in Korea. So training in States and also Australia. So I've been working on the autism genetics and dealing with these topics in early on with the colleagues. And after I moved to Korea, we also visit these issues, not just in the autism genetics, but also the sex differences as well. So, as you can expect, the autism or like neuropsychiatric topics in our country is quite underestimated, and also not many people really pay attention on these topics because we very focused on the cancers or other, the very the diabetes or Alzheimer's. But there are some the opportunities we can access to the data because there are the active research going on in Bundang Hospital of the Seoul National University around the 2010. So we access to this national level sample cohort. And we access to some genomic research and also the phenotypic data sets as well. Then we started working on these topics as well. And after a few years, we started the sort of research consortium we call the KARC, which is the Korean Autism Research Consortium. So this is the individual level investigators. They have the same goals, which is understanding autism and their biological background and also the clinical symptoms as well. So we building some the collaboration models, then we dealing with different topics on that. And also definitely one major topic is the sex differences in autism in Korean families as well. So, sex differences in Korean autism, we observed they're around 5 to 1, which is a little bit higher than the ratio we see in other courts, but not really big differences. But in terms of the medical insurance record, we see around the 61 ratio. So, clinical data set, at least we can, uh, you know, collect some samples from the milder background of the clinical symptoms, the female patients. And this data set, we also see a bit of the population background as well. So in 2011, there are the population level screening, so which is the not really clinical samples. This is the survey based one to the entire schools in one city. We call the Goyang. And we found around the 2.6% prevalence, which is quite high. And also this is quite similar level in states as well. And the ratio is about the 2.5 to 1, so which is quite low because these are not the clinical samples. These are the very survey based. So much higher numbers of the females which not diagnosed for autism, but they, captured in this survey. And this is one implication we can think of the clinical sex gap in terms of the ascertainment. And also some of the delayed and milder presentation in girls. So if you think about the clinical samples, so when it comes to the clinical visit, you have very obvious patterns in terms of like intellectual disabilities or very severe developmental delays. But this case, we can think of more the milder presentation or the symptom presentation is a bit late than male. So that also another implication we can think of in the female autism. And also we also looking at various co-occurring conditions. So the males and females has a different levels of symptoms in terms of the anxiety, depression, and also other comorbid conditions like eating concerns or the behavioral concerns as well. So these things also we narrow down with a very detailed phenotypes as well. And also, one major topic, especially I'm actively working on, is the genomic sex difference. So, when we're looking at the level of different genetic variants in autistic people, we also see different levels of the genetic variants across the males and females. So that would be the major topics we're going to discuss in later. This is the entire the pictures we are working on now. So the scales is around we have around 1,300 families and individuals around 4,000 people. So this is the matching to um, uh, the genotype and phenotype information we see in a European cohorts like Safari Foundation's SSC or the Spark dataset or the ISAC from the European dataset as well. So I mean, this is way smaller than those European dataset, but in terms of the compositions of genomic and phenotypic data set, we are quite confident we can test many different levels of information. So, which is really good starting point. Okay, so the KAIC, the consortium is basically harmonized by different levels of scientists. So it's including the clinicians, but also neuroscientists, which is working on a very molecular level, but also working on genomics and data levels as well. And as I mentioned earlier, and also emphasize again, this is not really the national level funding. This is the individual the PI's funding, and we harmonize and collaborate together. So we share the data and we share the idea, and we always trying to the best to come up with some idea about we can solve some problems. So when we design the experiment, when we design the some observations, we always discuss and also trying to find best research we can answer that question. So we working on this topic, and also we're trying to make some collaboration with other international collaborations as well. So we're trying to connect with other experts in other domains in other countries. So we use the different human nodes to connect with other expertise as well. So that's why we can work with other global consortiums like an ISAC or SSC, Spark, ASC. All these consortiums, we also see the same data set as well. Okay, so this is the details of the genomic data. So we are basically having the data set mostly from the whole genome sequencing, Illumina based, but also recently we used much advanced data set, which is the long read sequencing, mostly by PacBio, but also a bit of exome sequencing data as well. And we collect the phenotype data as well. So this is really big effort from the clinical team. So we are using the diagnostic tools like a very conventional tool, ADOS, ADIR, but also we measure the behavior ratings, so CBCL or the comorbid conditions as well. And also the beauty of this phenotypic data, so we're not just collecting the autistic individuals, but also collecting siblings and parents as well, because they have some very important domains we can think of the genetics and also phenotypic diversities as well. Okay, so back to the autism phenotypes and distributions as well. So as you can expect that the autism is one of the clinical diagnoses, but this is not really the binary condition. This is more like a continuous distributions we can see in our real worlds, right? So some are very severe and very strong autism presentation, but some are mild and some are not diagnosed for autism, but they quite, similar, traits to autistic individuals, and some are not, right? So this is very continuous distributions. And these distributions is basically composed by different levels of the factors. Now, I wouldn't say the risk factors, but the risk factors like a genetics, like a environmental factors, but also other common variants is naturally occurring in our DNAs. And in terms of describing these distributions, we need to use the various levels of genetic information. So when we're describing the distribution, some very severe phenotypes that is affected by one single factors, which is the de novo variant. Now we know about very strong and very reproducible way we can think of the autism factors. So this is the genetic variation occurring in a father's sperm or mother's eggs, and that transmit to the kids. So that's why it happens to the families which have no background in autism. So that is we call the monogenic factors. But some of the cases we can have the factors by different, the bit less rare than the de novo mutation, and that we call the rare variant, which is inherited from the parents to the kids, and that would be the monogenic case, so one single variant enough to contribute to autism, but also that will have the combination factors, so we call the oligogenic factors. Another things we can think of is the many different genetic factors, accumulating in one individual. We call the polygenic. This is the called by the common variant, and everyone in this room have these factors as well. So all of these factors is actually how we combine these factors in terms of our DNA. Then that will contributing to the place where we're sitting on these distributions as well. And these, factors is actually the very separating entities we measured over the decades. But now we have the technique called whole genome sequencing, and one technology can measure everything. So we use this method to measure the information. So very early observations we made in the Korean with the Korean data set was we published in Genome Medicine a few years ago. The first thing I have to look at very early point was that the Korean autism data set is really representing and really similar to the autism genetics. So first thing I can test with very reproducible factors, which is the de novo mutation. So when we're looking at the de novo mutation, especially the truncating the proteins, we well know about their distribution, well know about their frequency. So when we measure the autism patients versus unaffected controls, we see differences about 3 to 4 times higher in autistic individuals, and that is the observations we see in the European court SSN's part. If you're looking at the Korean patients, we also see the same patterns in both groups. So they make sure that the de novo mutation and also the autism, the collection by Korean clinicians is actually working, and we can move on to the next topics. So then we measure the polygenic scores, which is, as I mentioned, that by the common variant. So we use the autism GWAS, then build up some the scoring method. European cohorts, you can see the distribution from the very general population to the autism parents to autistic individuals. You see the deviation from very less autism risk to the more autism risk. And we see the same patterns in Korean data sets as well. So we use the general population data set and sibling the polygenic scores and parents and autism cases as well. So this is also ensuring that the whole genome sequencing and also polygenic method is working on in Korean data set. And also this is really representing the autistic the patient collections. Okay, so then we're looking at the sex differences. So there was another questions we majorly working on at the time. So if you're looking at the individuals, autistic cases with the intellectual disabilities without intellectual disabilities. So if you see here, the Korean patients have, especially the female cases with the intellectual disabilities, they have higher de novo variants compared to male cases. So which means that if you collect some autism patients in the clinic, likely have the very severe phenotypes than males. Otherwise, they're not really coming to the clinics. So that is one the things we can think of the ascertainment in autism diagnosis and also the research. And this pattern is already known in the SSC cohort, which is the state data set European cohort. That's also seen higher female cases in here. Spark is a little bit different. Spark is the mixed between the clinical samples, but also they have the survey-based samples as well. So they have bit less portion than the severe female cases. That's why I see quite similar rates in terms of the de novo mutation here. And this pattern is not really, distinct in the autistic cases without intellectual disabilities. They're also supporting the idea we discuss in here. And when we are looking at the polygenic scores in terms of the severity, so very severe group to mild group to the normative groups, you see the differences in terms of the polygenic scores. The very higher polygenic scores in severe groups to very lower level here. It's very similar to we see in the SSC cohort. But interesting stuff is that when we compare the female and male, that is the male has the higher the polygenic scores than female, which is the opposite to the we see in the de novo protein truncate mutation. So that would be we can think of that the different the sex difference, the genetic factors we can think of in here. And we expand this idea with the siblings and parents as well, because they have the sort of indirect indicator of the inheritance and genetics as well. So they forming sort of the genetic backgrounds of the families with autism individuals as well. So when we compare the siblings, which are not diagnosed for autism, when we compare between the female and males, you see the female, the non-autistic female siblings, they have higher polygenic scores than the male siblings, which means that they have greater genetic contributions than males. But interestingly, when we compare the phenotypic scores, we see the males have higher the phenotypes, so more severe phenotypes. I mean, that severity is not really reaching to the autism diagnosis, but in terms of the range of the unaffected people, the males have bit higher severe phenotype than females, which means that even though they have lower genetic background in terms of the common variant, but they really sensitized to in terms of the phenotypic presentation. And interestingly, that is also seen in the parents as well. So when we compare the father and mother, mother has likely have the higher polygenic scores, but in terms of the phenotypic levels, the father has the severe phenotypic presentation than mothers as well. So this is also the indication of the genetic factors also affecting on sex in different ways and also different levels of the sensitization as well. And so this is sort of the genetic layers we can think of monogenic and polygenic. So we really want to dealing with the genetic factors between these two, which means that the rare range, which is the very population specific, very specific to Asian, very specific to Korean, or very specific to European, and how these things is really affecting on autism, but also the different sexes in autism as well. And this is, we thought that the Korean autism data is really good point to discuss this topic. So we use some statistical model, which is we looking at the co-occurrence of the rare variants. We get one from the mother, one from the father, and how these things is really the frequently observed in certain populations. So we compare the Korean data set versus European data set. So we see some, the variant and gene pairs really sitting on the on this rare variant situation. And interesting stuff is that when we're looking at the de novo variants, the genes mostly working on the chromatin or the synaptic genes, we also see sort of those genes by rare variants as well. But rare variants, the genes they also see in the cytoskeleton organizations as well, which means that when we having the neurons and their differentiation, the genes like cytoskeletons or microtubules, they actively working on the neuronal circuit, and these things is disrupted by the rare variants very specific to the populations. And also, when we're looking at the sex differences in terms of these genetic factors, we also see the male bias as well. So when we're looking at the co-occurring, the rare variants we see in the cytoskeletal genes, these are the highly enriched in males compared to females as well. So this implicating that the male sensitization of the genetic factors it's not only happening in the common variants, but also happening in the rare variants as well. So these things we can work in further. Okay, so based on this evidence, we can think of where we should go further. So one thing we have to do with a high priority is that we need more females. We need more female with the autism, but also their siblings as well. So then we can see the entire sex biology in terms of the autism and autism neurobiology as well, and also how these the genetic factors and biological factors is really affecting on different sex and the autistic individuals as well. So that's why we're looking at the very longitudinal trajectory from the childhood to adulthood. But this is it's really depending on the funding continuity. So, but this is the things we can think of in further. And also linking the clinical genomic and EHR data. We are very fortunate to linking this data set in our consortium, but also there are many pieces of the information in our country, so we have to think about this way as well, and also the open data, and we make sure the standards of the entire the levels of different data as well. And also, not just the data set, we're also trying to put some validations as well. So, for instance, like molecular mechanisms of the sex differences, like a biology. So, one gene, like an CHD8, can also affect in autism different way in males and females as well. So, these things also we need to put in a future agenda to look up some molecular understanding and also drug development as well. So this is the recap of the entire topic. I think I'm running off the time. So we all talking about the sex-aware science and also sex-aware policies. So that's why we're implementing all of this idea into the research strategy as well. But this can be really based on the scientific evidence. That's really important thing. So scientists should actively working on with their evidence as well, not just the pieces of information. So that's the things why we are dealing with these topics in terms of the autism and developmental delays as well. So this is end of my presentation. I really thanks to some folks like Hee-Jung Yoo at Seoul National University and also Eun-Jun Kim at IABS. And also we have very strong international collaborations. Donna Walling, which is an expert in sex biology of autism. So she really helped us our project in very early point. And also thanks for your listening. I'm happy to take questions and also discussions as well. Thank you.